Welcome everyone. I hope you can see my slides and you can hear me. And so today I will go a little bit into what I have been doing over the past years, and in particular I was and then I'm still working in this field of optical DNA mapping, where I work with barcode analysis and I develop some tools. And so I will mainly not go into particular details and formulas of the things we're developing, but I will try to show some figures and maybe motivate of what I'm doing and why is it fun. First of all, you can see the view that I usually see every evening from my window in Lund in Sweden, where I'm currently situated. I'm living on the 9th floor of a nice building, and every day when there's sun, I can go away from my computer and look at the beautiful sunset. But while I'm not busy at home, I'm busy at the department. And so I'm, as already introduced, I'm in Astronomy and Theoretical Physics department in Lund, and we have this computational biology and biophysics group, and in this group we mainly work with nanochannel-based optical DNA mappings. So I was introduced into these activities by my supervisor, Tobias Ambjörnsson, and since I joined the group in 2016, end of 2016, we have written now some papers, and I will briefly go through of what is this optical mapping, what are barcodes, and what I have been doing, and maybe a little bit about what I plan to do in the future. So to begin with, maybe I thought it's nice to show a typical workflow of optical mapping assay. So this is from a paper which was written just about the time when I joined the group, so I didn't really participate yet in this particular project. But this project was related to rapid identification of intact bacterial resistance plasmids using optical mapping. So here on the top you see that there's some bacteria with some circular plasmids inside, and then there is this nice workflow that first extracts and stains plasmids with yoyo and natroxen. So this gives us a fluorescent pattern on the DNA, and this is then inserted into a nanofluidic chip. So in this nanofluidic chip, we move the intact plasmids from the microchannel into nanochannels, and then in the nanochannels they are in the nanochannels the DNA is extended, and then over time, since it's exposed to light, when we do fluorescent microscopy, the circular DNA breaks, and then we have linear DNA. And then the experimentalists start taking images, and from these images we get a chymograph, and the chymographs are basically the thing that I usually work with myself. So that's how our collaboration works, that we have a group in Gothenburg mostly that produces the experiments of various for various DNA and for various reasons, motivation, and then here in Lund I develop tools and methods of how to deal with these chymographs and barcodes. Right, so this is a very short outline of how a typical workflow would look like. And I have participated in developing quite a few of these different workflows. So maybe the first workflow is for image processing, and each of these tools have a nice 3, 4, 5 letter name. So so I have worked on developing image processing tools for this optical mapping. Then there are some things related to consensus barcodes, quantic assembly, alignment to some theoretical sequences of barcodes, structural variations, and also sparse barcode localization. So I want to mention here that most of my work that I do is for densely labeled barcodes. And this is maybe less developed field at the moment than the sparse labeled optical mapping. But it's still nevertheless quite interesting and growing field, and there's always new things happening. So, and I will go maybe a little bit through each one of these tools. So the first tool is the image processing. So here we have some tool called DBM. So this is a screenshot from application of this tool. And as the first step here, we use computational methods to detect where the molecule is in the image. So it's this red box that you can see in the screen. And once we have detected this red box, then we can extract a chymograph. And usually our experiments are over a number of time frames. So here on the x-axis you have the extension of the molecule. So this would be roughly 200 kilobase pairs, and on the y-axis you have time frames. Right, and then once you have this chymograph, maybe the very first application one can think of here is to measure the extension of these molecules. So for this one can calculate the edges of the molecule. So this, for example, could be done by fitting some kind of sigmoid functions on this chymograph. And once this is done, another thing that we usually do is to align the chymograph. Since you see now there's quite a lot of positional drift in between time frames. And we have some different tools that we have developed for aligning the chymographs, and then an aligned chymograph would look something like this. So it's you can see that the features are much more visible, and this aligned chymograph is then what we sometimes call barcode after we take time average over all of these time frames that you see. Right, and then, so as I said, we usually use like a number of time frames, but then a question is how many time frames we should use. Should we use 1, 2, 100, or 1,000? So in practice, taking more time frames takes more time, of course. So in this paper that we wrote in 2019, we actually did an experiment on a couple of bags from human genome. And we wanted to look how much the Pearson cross-correlation coefficient improves as we take more time frames, and then we take the average. And in the end, we decided that roughly 10 time frames is enough to get quite good results. And of course, the more time frames you take, the better you get. But if you take 100 or 1,000 time frames, then there's not so much big difference anymore. Right. And so once we have these chymographs, and we get barcodes from the chymographs after aligning, let's say, 10 time frames, then the next tool we can use is so if we have a number of these molecules imaged through our fluorescent microscopy, then we can take a consensus out of these different plasmids in this case. And so plasmids are circular, and we can put all of these in this tool that you have here again a little screenshot of, and then this tool runs a simple computational algorithm to get a hierarchical cluster of these barcodes. And here I have an example that I run on a linear molecule. So it's back from a human genome from the 22nd chromosome. And as an output of this, we get of this method of consensus barcodes, we get something which I call a Pac-Man map, because it looks like a little Pac-Man from a game. And you can visually see in this circular diagram the features of each particular different molecule. So this is slightly different from chimograph alignment, because in the chimograph alignment we had only a single molecule over different time frames, and here it's still the same position along the genome, but you have different molecules, and then you again increase the contrast of your final barcode when you use these. And so why is this useful? So we used the consensus barcodes in the first paper I wrote in the beginning of my PhD, which was related to making a quantic assembly using densely labeled optical DNA barcodes as a scaffold. And in this case, in this project, we had Illumina sequences as context. So we got context from Illumina sequencing, and then we didn't know how they are arranged, and we used this consensus barcode to actually place these contexts in order. And so what we found from this study is that the longer average context size is, the easier it is to place these context sequences on the consensus barcode, right? And so this was an interesting study and application of optical DNA mapping, and of course we don't get perfect results that we cannot map all of the small contexts, because we have some length threshold, because if you have very small of these barcodes, then you don't have enough features to give a significant match. Another interesting thing here was that we used combinatorial auction approach for placing these contexts on the optical DNA map. Yes, and so that was if we have a consensus barcode, but a different tool that I developed was called HCA, and this was mainly used for placing individual single molecules on the human genome. And in this case, so we don't know if, usually in practice we wouldn't know if the molecule is from a particular place on the human genome, and so then we would run a lot of experiments, and then we would place all of these molecules on the theoretical barcode, which would be generated from theoretical sequences, and then we would look if there is some cluster of points on some cluster of positions where you actually get placement of these individual barcodes at the same position. So this is an example of a plot of the software. So on the top left, you have PCC scores, Pearson cross correlation scores for each of the barcodes. On the top right, you have the positions. So maybe you see in the right-hand side of this plot there's some cluster, so that was actually the correct position for all of these barcodes. And on the bottom left, you have the best match between theoretical barcode, which was generated from theoretical sequence, and the experimental barcode. And on the bottom right, you have, again, the circular plot comparison between theory and experiment. And then, so we used this tool in a paper on enzymatic-free optical DNA mapping of the human genome paper. And so here's an example of 5 different bags that we placed on different chromosomes. And also in this study we used the p-values to evaluate if the match is significant. And so there was an interesting thing here that along the human DNA there's a lot of positions where the maps might match significantly, even though they're not. Even though it's not the correct position, because there's all kind of structural similarities if you look at the human genome. So we found out that we had to use quite a small p-value threshold to be able to get the number of false positives in our alignments to be quite small. But this decreased the number of useful molecules maybe to 20%, but so it's maybe a fivefold loss. But then we had much better guarantee that what we have mapped was mapped to the correct place. So that rose almost to 100%. And so we further evaluated then how well this densely labeled optical DNA mapping approach can map barcodes on the human genome. So we made a simulation where we looked at what is the percentage of human genome we could map if we just took kind of random 200 kilobase per barcodes and placed them on the theory, like 2 random barcodes with some, of course, from if you run experiments with random fragments. So we found that in practice we could cover basically 96.8% of the genome. And then as the continuation of this, there were other people in the group were trying to develop dual label methods where we would combine sparse and dense labeled methods to actually improve this alignment and maybe be able to cover the whole 100% with both approaches. Right, and so once the study was done, we continued with another application where we wanted to find species of bacteria using optical DNA mapping. And in this case, we again developed a workflow with collaborators. So in particular, it was former PhD student Wilhelm Muller, who was doing really great things here. And so here, again, we started from all this extraction from urinal sample and then labeling and putting the molecules in the nanochannels. And eventually the goal was to identify the species of bacteria. And for this, we developed this method for finding discriminative profiles yeah, so it's a rather interesting approach, I think, and there were then maybe I included a few examples of how this method works. So here you have 3 different E. coli isolates, and you can see how their barcodes were placed on the theoretical barcodes. And in this case, for each of these, we had a certain number of barcodes that were imaged using this optical mapping. And we found that in each of these cases, the mappings were discriminative. So in the first case, it was 34, second 11, and third one 20. So that was a quite nice output. And then there were more complicated things that we were trying out. So we also tried bacterial mixtures, and we were trying to see how successfully we can identify correct species when we have mixtures of different bacteria in the same sample, and also now as a continuation of the study, we're also working on subtyping of subspecies of finding subspecies of bacteria. But then the methods are a little bit more involved as well. Right, so that was cultivation-free typing of bacteria, a little bit insight. And the last 2 things that I will briefly talk about are not yet available in published literature, but the first one of these that I will mention briefly is finding structural variations in these barcodes that we work with. And in this case, you have, so here I just show a visual example how a tool that we were developing, which is a hidden Markov model-based structural variation detection tool, works on 2 barcodes. So you have here on the top query barcode, on the bottom reference barcode, and then we want to find if there is some structural variation in the query. And actually, so there's these boxes of 2 different colors that map to particular regions from one barcode to the other. And actually, you can see here that there is repetition in the first barcode, and it showed us overlapping part of the second barcode on the bottom. So this was the tool that I was developing for detecting structural variations. Right, and I think this is going to be soon available in a publication. So I'm quite happy about that. Right, and then so this is very quickly about detecting structural variations. So, and note here the barcodes that we have here are roughly 250 kilobases long. Right, and then the last thing that I was working on and still I'm working on. So this is more related to sparse label barcodes. And in this project, we want to kind of get high precision fluorophore localization on the barcodes. So here comes the name from HPFL. And so here I have been developing a workflow which starts from image processing. So here we have a processed image from Iris machine. So it's one of the commercial machines for running optical mapping experiments. And here in the image, in red lines, we have detected the nanochannels in which there are some molecules. And once we have done that, we can extract the chemographs So here you have a picture of all the different gamma graphs that you can get from these experiments. And in this method, what we want to do is then we want to take a specific region on one of the gamma graphs and see how well we can locate the positions of atoms or the positions of fluorophores in these maps. So for this, I have an example here of this interesting part of the molecule. So you see here a gamma graph of a repetitive region of the DNA molecule. And here we're developing some nonlinear optimization-based method that detects positions of atoms in each time frame. And then we, since we know that this is a repetitive region, then we can compare actually using similarity scores, such as, for example, checkout index or precision or recall, to see how well we get the positions out of these maps. And also in the study, we so this is like an example of a single time frame recovery of for these barcodes. So in blue here you have the raw signal of the barcode, and then the red is the positions of the detected peaks, and then you have like these recovered Gaussians in black. And also in the study we have developed some simulation of these barcodes, because so we developed simulation of these gamma graphs, so we can better estimate how well our method performs when we have, for example, photo bleaching, photo blinking, and also when we have different noise conditions. Right, and so that's the main things that I've been working on, but there were also some other things that we are interested in the group. And it's one thing is, for example, assembling the chromosome using this dense optical mapping. Of course, there's already many tools to do this in first parts labels, but for dense labels, it's maybe a bit more challenging. But we still nevertheless want to be able to successfully assemble some chromosomes. And also we want to find structural variations on chromosomes using these dense maps. There's, I think, already submitted and almost accepted paper on dual labeling, which I already mentioned a little bit before. And we also always try different measures and methods for how we can align the experiments to theoretical barcodes. So in particular, I've been working quite a bit on this matrix profile method, which aligns sub barcodes of one long barcode to the theory. So this is also helpful in detecting structural variations, for example. And then we might want to combine the tools that we already have with some other commercial tools, like Sapphire, which is a new tool for optical DNA mapping, rather new tool. And then also we want to always improve, for example, the theoretical barcodes that we generate. And I guess there's many more directions that one could go in, and maybe I'm more concentrated on algorithmic development, since I stay here with my computer in the lab, in lab which is an office, and then we have collaborators mainly in Gothenburg, in Westerlund lab, who then help us out with experiments. Yeah, so that's basically what I wanted to share today with you, and thank you for attention.
Thank you so much. I really liked that you brought up so many ways to use the research that you've been doing and how applicable it is already. When I studied up on this, it seemed like there's a long way to go with the limitations of, for example, like the chromosomal DNA and stuff. But what you really showed here today is that a lot of things can be used with tools and methods that you've already developed and helped doing. So thank you a lot for this. Very interesting. I have a few questions, if you don't mind. And,
yes. Go.
I was wondering, in your algorithm, you didn't go into specifics today,
No.
but a very important part of the algorithm is the non-overlapping contig constraints. I was wondering if you would want to speak a little bit about this constraint and how it affects your analysis, and if it's something that is specific for your tool or if it's
Yeah, so that would be so I guess that was particularly related to contig assembly.
Yes.
Yes. So in this particular tool, we used the combinatorial auction approach, which had a non-overlapping constraints. That's true. But this is not the issue, because also the context that we get out of the sequencing are also non-overlapping. So
okay.
So the method then works for that. Yeah.
That's really nice.
Yeah.
Okay. And I was wondering also, in the cultivation-free typing of bacteria that you mentioned in your presentation, how much time do you think can be saved with using this experimental approach instead of the alternative bacterial typing methods?
This is a bit hard to tell me, because this method is also like like the time that it takes to get from the sample to the final output. It's also all the time changing, right?
Yeah.
So now I don't recall a particular number of how much time it takes to get from taking a sample to get the output. So I think I cannot say how much time it takes.
Yeah, thank you for your answer. Also, in your lab, you focused to work with densely labeled DNA maps. Obviously, both, as we learned. Would you say that densely labeled DNA maps have an advantage over sparsely labeled? In what type of applications do you think there's an advantage of having the densely labeled?
So I think the particular advantage could be, so we also have places along the genomes where sparse labels might not be covering the whole genome, and then you would need to be playing around with different sparse labels.
Yeah.
Yes, and also when we, for example, look for structural variations, this dense label gives us more of like the profile of the feature, while in the sparse label we would have a position, or we wouldn't have the position. So here we have a little bit more information on also on the shape of the of the barcode. Yes. So that's
I see that we have question or two actually in the chat, and the first one is: do you think your method can be combined with a long read sequencing technique?
Yeah, so I think that is possible, but there's not so much research done so far, at least for the combining the dense label barcodes, the long read sequencing, but there are some articles already that tried to combine long read sequencing with sparse labels. So I think it should be possible to combine the two methods. Yes.
Thank you. And the second question: can it help with phased methylation analysis, do you think?
Yeah, well, here I'm not very familiar with phased methylation, so I don't think I can answer that at the moment, but yeah.
Okay.
I'm not aware that something would be done in that. Yeah.
Yeah. Once again, thank you so much.